Hello students, welcome to EPG Pathshala. I am Dr. Rita Rath from Dayal Singh College, University of Delhi. Today we are going to discuss about a module Trichinella spiralis under the paper Parasitology. The learning objectives of this unit is the unit will help us to understand the medical importance of Trichinella spiralis, identify the male and female worm from its morphological characteristics, explain the importance of hosts in the life cycle of Trichinella spiralis, diagnose the symptoms of the disease caused by the parasite, understand the genomics and proteomics of the parasite so that enable us to design more accurate diagnostic, preventive and curative measures, and to suggest various methods for the prevention and control of the parasite. The parasite can be classified as it belongs to kingdom Animalia, phylum Nematoda, class Adenophoria, order Trichocephalida, family Trichinellidae, and the genus is Trichinella and the species is Spiralis. A brief introduction about the parasite. It is a nematode parasite which is able to infect rodents, pigs, horses, bears, and humans. It is responsible for a disease called as trichinosis. It is also commonly called as the pork worm due to being commonly found in undercooked pork products. One of the most widespread and clinically important parasite in the world. It is the world's largest intracellular parasite. History of Trichinella spiralis. Trichinella spiralis, which causes trichinosis, was first observed in 1821 in the muscles of a patient at autopsy done by James Paget. Richard Owen in 1835 described the encysted larval form in muscles and named it Trichina spiralis. Virchow discovered its life cycle in 1859. The name Trichinella spiralis was given by Rayleigh in 1895. Trichos meaning hair, ella meaning diminutive, and spiralis refers to the spirally coiled appearance of larvae in muscles. It occurs in humans both as an adult in the intestine and as a larva in the tissues, usually the muscles. Trichinella spiralis shows a cosmopolitan distribution in temperate and equatorial climatic zones. Several recent reviews summarize the presence of trichinellosis in individual countries such as Argentina, Hungary, China, Mexico, and Greece. Furthermore, due to political and economic changes in southeastern Europe, a re-emergence of trichinellosis has been reported in countries of this region, and recent human outbreaks have been reported in Germany, Italy, and United Kingdom. Human trichinosis has been reported from India in 1996 when the first case was seen in Punjab. Geographical distribution of Trichinella spiralis. Species of Trichinella inhabit a broad geographic range from the Arctic to tropics. However, distributions of individual species are more restricted, with encapsulated species generally demonstrating adaptations to colder climates than non-capsulated species. With the exception of some non-capsulated species, noted major host groups of Trichinella species are domestic and sylvatic swines, synthetropic animals such as the brown rat, the armadillos, cats, dogs, and a broad range of sylvatic carnivores. It has been passively imported into most continents due to its high infectivity to swine and synanthropic rats. Habit and habitat. Adult worms remain buried in duodenal or jejunal mucosa of pig, rat, and humans. The encysted larvae are present in the striated muscles of these hosts due to the fertilized females discharging embryo into the circulating fluid and reaching the muscles where they ultimately remain uncysted. The adult, it is one of the smallest nematodes infecting man and is white in color. The male is 1.4 to 1.6 millimeters in length and 0.04 millimeters in diameter, whereas the female is much longer, measuring 3 to 4 millimeters in length and 0.06 millimeters in diameter. In males, the copulatory sheath and spicules are absent, but on the either side of the tip of the tail there are two conspicuous conical papillae termed claspers that it uses to hold on to the female worm during mating. The females are viviparous and release first stage larvae into the intestinal mucosa. Larvae. They measure 100 millimeters in length and 6 millimeters in diameter and remain encysted in striated muscles of the host. It then grows inside the cyst till sexual differentiation becomes 10 times its original size from 100 micromillimeters to 1000 micromillimeters. Reaches maximum size by 35th day and normally only one larva is present in one cyst. Encapsulation of larva starts by about day 21 and completed in 3 months. An ellipsoidal lemon-shaped sheath develops as a result of host tissue interaction around the tightly coiled larva. The long axis of the capsule is parallel to the muscle fiber. Calcification occurs in 6 to 18 months. The muscles which are heavily parasitized are the diaphragm, intercostal, pectoralis major, deltoid, biceps, and gastrocnemius muscles. There can be 1000 trichinella per gram of muscle. Trichinella spiralis has a direct life cycle, that is, it completes all stages of development in only one host. The larval forms are encapsulated as a small cystic structure within the infected host. Humans become infected when they eat improperly cooked trichinella infected pork or any other meat. Due to the pH in the stomach, the larvae are released and migrate to the intestine. They burrow the intestinal mucosa, mature, and reproduce there. The larvae can then gain access to the circulatory system and migrate around the body of the host. This causes fever and pain brought upon by the host's inflammatory response. Also, migration to specific organs or tissues can cause myocarditis and encephalitis that can result in death. The larvae is freed from its nurse cell and enters the mucosa of the small intestine. Larvae grow, molt 4 times, and young adults copulate within 32 hours of infection. The females give birth to live larvae while tunneling the epithelium. Females die after 4 to 16 weeks. The larvae, after hatching, enter the blood vessels and are carried throughout the body. When they pass the skeletal muscle, which they normally prefer the active muscles, they leave the vessels and penetrate the muscle and the muscle cells. The larva enters a muscle cell and leaves as an intracellular parasite developing in the cytoplasm of the host cell. The larvae manipulates the host cell to its needs, probably by secretion of suitable effector proteins, the molecular mechanism of which is not well understood. The end product is a nurse cell. The fine net of blood vessels forming around the nurse cell by a process known as angiogenesis. The host cell then loses its myofilaments and several additional subcellular changes occur. Both the host cell and the worm are enclosed by a collagen capsule. Once the infected meat is taken in, the larvae are released from the nurse cells in the stomach due to its pH. The larvae then enter the small intestine. Adults mature and live in the small intestine. The females shed newborn larvae that enter the lymph blood. The newborn larvae are carried throughout the bloodstream. They may infect the CNS or the central nervous system and damage it, or the heart, leading to heart failures, or may enter the skeletal muscle cells. In the skeletal muscle cell, the larvae matures. A nurse cell larva complex is formed, and the normal or the red and calcified nurse cell larvae complex is encysted in the muscle tissues. Trichinosis or trichinellosis. The disease is called trichinosis or less commonly trichinelliasis or trichiniasis. Manifestation varies from asymptomatic infection to an acute fatal illness, which is rare. The clinical feature is classified according to the stage of the life cycle of the worm. There are two phases. First phase is the stage of intestinal invasion or enteric phase, also known as the incubation phase. This occurs in the early stage of infection, normally 5 to 7 days, during which the larvae grow into adult worms and the female begins to discharge larvae into the circulation. Symptoms are gastrointestinal nausea, diarrhea, abdominal cramps, and sometimes vomiting. This is diagnosed as acute food poisoning, particularly when it occurs in groups of people who have taken the same food. In some, constipation is seen instead of diarrhea. The onset of illness may be from 2 to 30 hours after the ingestion of infected food. The stage of larval migration or the muscle invasion or migratory phase. This occurs during the release of larvae, their migration, deposition, and encapsulation in muscles. This occurs from 7th to 10th day and covers a period of 4 to 16 weeks. The chief symptoms are fever, edema of face, swelling and weakness of affected muscles. Eosinophilia is a constant feature. Myocarditis and encephalitis are serious and potential fatal complications of toxemia. Then the stage of encapsulation or encystment phase. This occurs only in striated muscles, while in other tissues they degenerate and are absorbed. This lasts from 1 to 8 months after infection, and fever and other symptoms have subsided. After this stage, the cyst begins to calcify and may have permanent injury. The clinically disease is in light and 2 to 3 months in heavy infections. The second or the muscle phase, which leads to muscle aches, itching, fever, chills, and joint pain, and which begins after 2 to 8 weeks after ingestion. Also, there can be splinter hemorrhages under the fingernails and eye inflammation leading to conjunctivitis. Diagnosis of the disease. Clinical diagnosis is helped by the history of consumption of inadequately cooked pork or other meat. Correct diagnosis can only be achieved by demonstrating the muscle obtained either from biopsy or autopsy. The following measures can, however, be adopted. Number 1, the stool examination. Presence of adult worm in the feces is rarely possible. The second is the blood examination for leukocytosis with an ascending eosinophilia. The third method could be the serological tests such as complement fixation, precipitin, and bentonite. The fourth could be the muscle biopsy by third or fourth week of infection. Suitable sites for biopsy are the tendinous insertions of deltoid and gastrocnemius muscles. The fifth diagnosis could be done by a skin test, the intradermal injection of 0.1 ml of 1 in 10,000 dilutions of Beckmann's antigen causes an immediate erythematous patch. And the sixth method could be the X-ray examination if the cysts are calcified. Most trichinosis infections have minor symptoms and do not require any treatment, as all symptoms resolve without treatment. With more intense symptoms, thiabenzidole, also known as mintazole, can be used to eliminate the adult worms in the gastrointestinal tract. Albendazole, also known as Albenza, is another drug that may be used in some cases. The invasive and encysted larva forms of Trichinella species are treated by mebendazole, also known as Vermox. Inflation of infected tissues is usually treated with prednisone and is frequently used in combination with mebendazole. Control and prevention of the disease. The pork should be cooked thoroughly. Also, the flesh of bear, walrus, and other wild pigs need to be cooked properly. Proper meat handling and ordinary curing and salting of pork products does not kill encysted juveniles. Freezing is effective if carried out properly. The freezing requirements differ with the size of the meat. Pieces not exceeding 6 inches in thickness require 20 days at 5 degrees Fahrenheit, 10 days at minus 10 degrees Fahrenheit, and 6 days at minus 20 degrees Fahrenheit. Larger pieces, however, require a longer period. Quick freezing and storage for 2 days is effective. Cooking the wild game meat thoroughly also helps. Freezing wild game meats, unlike freezing products, even for long periods of time, may not effectively kill all the worms. This is because the species of Trichinella that typically infects wild game is more resistant to freezing than the species that infects pigs. The Trichinella spiralis draft genome became available in March 2011. The genome size was 58.55 million base pair with an estimated 16,549 genes. It was subsequently shown that the Trichinella spiralis genome is the only known nematode genome to be subjected to DNA methylation, an epigenetic mechanism that was not previously thought to exist in nematodes. The genotype and the phylogenetic position of Trichinella within the phylum Nematode. Molecular phylogenetics has defined three major nematode classes, which can be further divided into five clades: Dorylaemia, Inoplea, Chromatodoria, including Spirulina clade 3, Hylanchina clade 4, and Rhabditina clade 5. Parasitism has arisen multiple times during nematode evolution, and all major clades include the parasitic species. The model free-living nematode Sinorhabditis elegans is a member of Rhabditina clade 5, and T. spiralis is a member of Dorylaemia clade 1, making these examples some of the most distant species within the nematoda. The first robust and comprehensive analysis of the phylogeny and biogeography history of Trichinella was recently reported based on variation in several genetic loci. The basal Dorylaemia lineage, which contains Trichinella, also contains the free-living Mononchida, the plant parasitic Dorylaemida, and the entomophagous Myrmidida. These nematodes share features of early embryogenesis and small subunit rDNA sequences. The genus Trichinella is a monophyletic lineage in the Trichinellidae, which diverged 275 million years from the putative sister Trichurida. The 11 known species of this genus diverged into two distinct clades. Agreement for that divergence comes from both biological considerations. Trichinella clade 1 are encapsulated and clade 2 are non-encapsulated, and the genetic data. There are currently 8 recognized species or genotypes that comprise this genus. The species display diverse biological characteristics, the evolutionary significance of which has been recently extensively clarified. Some of that diversity translates into variable importance as zoonotic pathogens, with Trichinella spiralis having the highest significance. Trichinosis has re-emerged as an important zoonotic infection in various parts of the world, reminding us that control of this infection depends on persistent vigilance. Trichinella species display unique and biologically interesting complexity in interactions with host cells that they inhabit. Significant progress has been made towards understanding details of these interactions. Progress on transcriptomics, proteomics, and now genomics offers exciting prospects for accelerating advances in future research. The Trichinella assembly is 64 million base pair in length and encodes at least 15,808 proteins, which makes this genome substantially smaller than the prototypical nematode Sinorhabditis elegans. The nematode T. spiralis, the most common cause of human trichinellosis, is a member of clade that diverged early in the evolution of nematode. The lineage giving rise to genus Trichinella last shared a common ancestry approximately 275 million years ago, whereas the diversification of extant Trichinella species occurred as recently as 16 to 20 million years ago. The repeat content of Trichinella spiralis genome is estimated at 18%. The repeats have a low GC content, which is around 27%, and relative to the genome overall, 34%, and to protein coding regions, about 43%. T. spiralis lacks both the canonical SL1 transspliced leader found in most nematodes and the SL2 transspliced leader that is spliced onto transcripts from downstream genes in C. elegans operons. The number of predicted genes in T. spiralis is notably lower than 20,140 genes identified in C. elegans, even though the two genomes show similar repeat content and gene density. The National Human Genome Research Institute, NHGRI, called for the proposal for species that represent a position on the evolutionary timeline marked by important changes in the animal anatomy, physiology, development, or behavior. T. spiralis genome sequence was expected to contribute in several ways. First, it was to provide a better understanding of evolutionary biology by identifying gene loss or gain across the phylum Nematoda and clarify evolution of gene architecture, synteny or operons. Second, it was to help identify RNA genes and the regulatory regions. Thirdly, for better defining the proteins involved in nematode parasitism that impact health and disease and are relevant to both host-parasite relationship and basic biological processes. In 2004, NHGRI announced that the large-scale sequencing research network should receive financial support for sequencing the genome of the adenophorian nematode of clade 1, that is Trichinella spiralis. Proteomics of Trichinella. Proteomics tools are used to analyze excretory and secretory, known as ES products, in parasites. ES proteins are believed to have roles in formation of the host parasite complex and including changes in the host cells. Early studies involved surface labeling of proteins on 3 stages of Trichinella spiralis with iodine and examination by 1DE or use of 2-dimensional electrophoresis for identifying excretory-secretory proteins in infected muscles or muscle larvae. The attempts resulted in analyzing only few ES proteins in terms of their biological activity, due largely to problems in identifying individual proteins. Furthermore, 2 proteins found in the ES fraction collected from the muscle larvae cultivated in vitro lacked a typical N-terminal signal sequence, while secretion appeared to be mediated through the classical endoplasmic Golgi secretory pathway. Later studies used 2-dimensional electrophoresis and proteomic analysis to identify the excretory-secretory proteins from T. spiralis. The PMF data obtained from Malditoff mass spectrometry analysis of ES peptide spots excised from 2-dimensional gels was used to confirm the existence of 2 members of a family of nematode-specific proteins that have N-terminal signal peptides. Expressed sequence tags generated from the 3 life stages of Trichinella spiralis, that is the adult mature muscle larva and immature L1 larvae, improved technical capacity for proteomic research on Trichinella species. By coupling protein sequence similarity with signal peptide prediction, 345 Trichinella spiralis clusters were identified that had homology with predicted secreted or membrane proteins. The EST clusters supported interpretation of peptide mass fingerprint data obtained from two-dimensional electrophoresis analysis of muscle larvae ES proteins. More recent two-dimensional electrophoresis of ES proteins was coupled with MALDI-TOF and LC-MS/MS, enabling the most comprehensive identification of peptide spots from T. spiralis. Identities were assigned to 43 out of 52 ES peptide spots analyzed based on either the PMF data or de novo peptide data derived from LC-MS or MS. Interestingly, the 43 spots represented only 13 different proteins, indicating that there are multiple protein isoforms present in the ES, the most prominent of which are a serine protease, the 45 kilodalton antigen, group 43, and two unidentified open reading frames. To summarize this, Trichinella spiralis, commonly known as the pokeworm, is a nematode parasite which is able to infest rodents, pigs, horses, bears, and humans. It causes the disease called as trichinosis. It is the world's largest intestinal parasite. It has got a cosmopolitan distribution. The female is larger than the male. It has a direct life cycle, that is, completes all stages of development in one host. The humans become infected when they eat improperly cooked Trichinella infected pork or meat. The life cycle starts with the ingestion of raw flesh with cyst by humans. Then excistation in duodenum occurs, the adult worm develops, fertilization occurs and deposition of larvae in the intestine, and from there the entry of larvae into the circulation and distribution in striated muscles. Trichinosis is characterized by abdominal discomfort, diarrhea, nausea, muscle ache, itching, fever, chills, joint pain, sphincter hemorrhage under fingernails, and eye inflammation leading to conjunctivitis. It can be treated with thiabenzidole, albendazole, mebendazole, and corticosteroids. Infections can be prevented by proper cooking of pork or freezing. The genome size of Trichinella spiralis is 58.55 million base pairs with an estimated 16,549 genes, substantially smaller than that of the prototypical nematode Sinorhabditis elegans. Proteomic tools are used to analyze the excretory and secretory products in the parasite. Thank you.